This year, for the first time in our series of conference, Professor Hui Li sent us an email to present the idea about chimeric RNAs, and we found it very interesting. And the idea of this conference is we record the conference, and the conference is going to be posted in our webpage. So from time to time, you should go there because we are going to do this in all our conferences. It's some kind of TED Talks, but in this between family, okay? So thank you very much indeed, Hui Li, for accepting to be here today. He comes from the University of Virginia, and he's going to present chimeric RNAs as a new way to expand functional genome. Thank you very much, Li.
Thank you, Luis. Thank you for the invitation. First of all, University of Virginia, not many people know about it or not as familiar as Harvard or Princeton, but this is one of the best public university in US. And the campus was voted the most beautiful campus in US last year. It is also the only US campus that's listed on the World Heritage site. All right, so starting from something basic, gene is defined as the molecular unit of hereditary information according to Wikipedia. Now you like this because you love dogs. The central dogma of biology, we all know that the DNA genetic information flows to RNA to protein. Of course, we know reverse transcription. We also know alternative splicing. That's way to expanding this repertoire of transcriptome or functional genome. But I'm going to talk about chimeric RNAs, actually made of two fragments or transcript fragments from different genes. And in this recent review, we actually highlighted some of the findings on that topic where you could make chimeric RNA from our well-known mechanism like gene fusion in cancer, chromosome translocation. But there is also a mechanism like transsplicing could connect the chimeric the RNA fragments into chimeric RNAs, as well as a C-splicing of adjacent genes, and some people call it transcriptional read-through, but to avoid confusions, we call it C-stage, C-splicing of adjacent genes. That could actually exist in normal physiology as well as in cancer, and some of these behave into the chimeric proteins and as well as non-encoding RNAs, or results in differential expressions of the parental genes. Starting from cancer, because I'm trained as a cancer biologist, so the story really begins when we were working in a particular chromosome translocation and gene fusion event. And in cancer genetic field, we also know there were two dogmas. One is, dog lover, that chimeric fusion products is mostly due to chromosome translocation. That's one dogma, that you see a fusion RNA or fusion protein that's made because there's a chromosome translocation or rearrangement. And these fusions must be unique to cancer. And that's really the fundamental basis for a lot of molecular diagnosis of potential even therapeutic approaches, because we thought the fusions or fusion RNAs and proteins are unique to cancer. And when we were working on a particular chromosome translocation in an endometrium sarcoma, we found there's a fusion of T717 translocation, Jazz-I5, JJAZ, another name is SUSI12 fusion. That's also present in normal endometrium cells. So it was a little surprising. We see it at RNA level, not only by RT-PCR, but RNase protection assay. We can detect it at the protein level. But surprisingly, there's no abnormal DNA rearrangement. So there's no chromosome translocation that, as you see in the cancer. What happened is, it turns out to be a transsplicing event, where some of the exons of the Jazz-I5 go transsplice into some of the exons of the SUSI12 gene. And so, long story short, we proposed the model where you can have your traditional chromosome translocation and results in chimeric RNAs or chimeric proteins, but you could also happen by transsplicing, and that could happen in normal physiology. The interesting thing is the final product is actually exactly the same. So at the time we proposed the model that, well, it's not really we're the fast, but we all know that cancer is smart but not original. So cancer hijack or use a lot of machineries or mechanisms that you actually can see in normal physiology. Well-known example is the EMT. But through chromosome translocation, you may result in an elevated or uncontrolled way to express certain potential proto-oncogenes that may play a role in normal physiology. So with that in mind, actually, we also discovered this C-splicing of adjacent genes as another mechanism to produce chimeric RNAs. So with that thought in mind, we thought to ourselves, how are we going to discover more meaningful chimeric RNAs? And this is about 8 years ago when we started this, and the RNA sequencing was not widely available. So we took the approach of guessing. There are about 200 well-known chromosome translocation gene fusions in cancer. We asked the question, any of these could happen in normal physiology, and if it does, maybe it's a product of transsplicing. So what we did is actually, since if such a thing exists in normal physiology, it wouldn't be so abundant, it wouldn't be constantly be there, because otherwise these wouldn't serve as cancer markers. So they may only transiently express in certain cells at certain lineage under certain conditions. How are we going to find these kind of transient event? And we decided to follow stem cell differentiation approach. So for instance, if you take mesenchymal stem cell, put in the cell cultures, and give them different stimuli, you can get them change into or differentiate into bone, cartilage, muscle, fat, stuff like that. And so we followed the muscle differentiation lineage and was able to detect a classic chromosome translocation or gene fusion event, PAX3 FOXO1 fusion in rhabdomyosarcoma, type of rhabdomyosarcoma. And that is actually considered a signature of that type of pediatric cancer. But we could actually see that particular fusion being expressed transiently during the muscle differentiation. I won't go into any of the data since it's already published, but it's precise the myoblast stage. So such a classic cancer signature fusion or does exist in normal physiology, and it was only there transiently. If you don't look at right time, you would not see this kind of things. What does this mean clinically? Of course, it's going to mean that there may be false diagnosis. If you're trying to, especially monitoring residual disease, where you use a very sensitive assay like RT-PCR and see if the patient are free of the cancer cells, then potentially such a normal physiologically existing fusion RNA might be a problem for false diagnosis. Secondly is this idea may shed light on the self-origin for mysterious tumor. So like I said, the alveolar rhabdomyosarcoma has been debated many years for the exact self-origin. Some people believe it's a precursor cell, it's a mesoderm precursor cell. Some people believe it's myoblast. Some people believe it's a mature muscle but de-differentiated back to the certain lineage. But the evidence that such a classic signature fusion RNA exists in the normal physiology at certain time, shedding light on the potential self-origin for this mysterious tumor. And long and behold, we actually did more work on this topic, and particular, say, different time point of muscle differentiation. And on time point 3, we identified a lot of fusion RNAs that's exactly expressed as the cancer cell has. And this actually we can show, this is only part of the story, but almost all of the fusion RNAs you see in the cancer could later on be detected at that particular time of normal physiology. Again, I won't go into details, but we came up with the idea of using the fusion RNA profiling to actually connecting some of the biological samples and may shed light on some of that hidden connections. And this connection, you would not even see it with a whole transcriptome clustering because there's just too many, 50,000 transcripts are noisy, and the fusion RNAs tend to be more black and white. All right. So I was given an opportunity to explain some of these data to a bunch of Hollywood producers, and so using examples that they may understand, I use the, I use Robert Downey Jr. development. So this is what we consider normal development. And he got into trouble, put in jail 2 times for some drug usage. So that's called a tumor genesis. And so over here, you can say, okay, what's the self-origin for this bad guy here, right? It's not this one innocent, and it's not this one now is well-behaving, but it's this guy because of some features, like how many white hairs he has or how many wrinkles on his face. Those features may or may not contribute to the tumor genesis itself, but it works as fingerprints. to connecting these two biological samples. And so we believe this actually is the tip of iceberg, and we're utilizing these ideas to undermine a lot more connections between certain cell types as well as cancer. All right, so conclusion for this part one, chimeric fusion RNA does exist or do exist in normal physiology, and certain cancer signature fusion RNAs could also occur during development. Using the fusion RNA profiling may lead to new insights into mysterious tumors. All right, so after the RNA sequencing becoming widely available, of course, you can think of the genome-wide approach, which is just using the parent RNA sequencing, and that's exactly what we did with a lot of bioinformatics. Of course, I won't go into details, but when we talk about chimeric RNA, right, you think about the chimera, and the chimera is this Greek, mythology monster, has a lion head and serpent tail. Sometimes there's a ram head in the middle. We're not going to talk about three-way chimeras. It's too complicated. But, just chimeras in general. And so, the Greek hero, the Bellerophon, killed the chimera, with the help of Pegasus, right? Pegasus has two wings, and that made us think of parent RNA sequencing. So you need that. You need parent RNA sequencing. And you need a map, right? So in this case, the map is really Circles plot. So, for people who are not familiar with Circles plot, you put the whole genome on there, chromosome 1, 2, 3, 4, X, 22, X, and Y, and each line indicates there's a chimeric RNA linking the two parental loci. And so in this particular case, we did, I'd say about 190 wild RNA sequencing libraries and analyzing through normal donors samples. And we were able to detect a lot of chimeric RNA events, including embryonic stem cell differentiation, MCF10A, which is a non-cancer breast mammalian mammary gland epithelial cell line, as well as the mesenchymal stem cell differentiation to muscle. And we detect a lot of chimeric RNA events. Some of these are tissue-specific. Some of these are dynamic during the differentiation process. So this highlights that we are really underappreciating these hidden or dark matters in the universe until you find a way to find them. And then, we also did validations and proving they're existing. Some of these could be detected if you have a Western blot antibody that you can detect at the protein level. Some of these we did high throughput mass spectrometry and detecting the chimeric peptide, but not all of these are going to translate into protein. Some of these work as long-encoding RNAs or affect the parental gene expression. Recently, we finished, and I will just talk about the GTACs, of over 9,000 RNA sequencings comprising 500-ish individuals that's donated 53 different tissues on average. And we actually finished all these data analysis, and these, again, emphasizing from normal, or at least they tried really hard to get the non-diseased tissues. And this serves as a great resources for us to data mine the non-disease related chimeric RNA profiles. And from there, we actually find tons of such events. And we can even use the binary input of chimeric RNAs. So either there or not there, and do the t-SNP plot and separating different tissue types. So this, again, highlighting, and then the transcriptome analysis can do about the same, but here is really binary. It's there or not there, and we can actually do very good segregations in this particular case as well. So now the fun really begins. All right. When we say that the gene numbers can explain the difference, this is the audience. I don't have to say much about it because we all appreciate alternative splicing, but we all share about 20,000 genes, right? It doesn't matter your worm or the Drosophila, and then a lot of plants has actually, twice as bigger gene numbers as us. So gene number can't really explain all the situations. Can chimeric RNA provide some of or at least work as an alternative mechanism to expand some of the functional genomes? In the lab, we actually are very interested in finding such weird examples. And so, for instance, tissue-specific fusion RNAs or sex-specific fusion RNAs. Apologize, I cannot use gender. Gender describes different things. I'm going to have to stay with sex. And then fusion RNAs correlating with age, race, and different things. So I won't have time to go into all these, but just give you a few examples. I have a visiting scholar who delivers babies, and she come to the lab, and she only said, I want to work on placenta. So fine, placenta, she actually did the sequencing, find some placenta-specific chimeric RNA, and it does, this particular one is truly there. And when she actually looking at the fusion RNA through the tissue panels, we can detect it in the placenta, but not in any of the other tissue types. But when she look at the parental gene, it's basically expressed everywhere. So suddenly, the parental gene are expressed every tissue, but suddenly the fusion RNA becoming a tissue-specific phenomena. And the next thing she did was she was actually looking at the placenta trophoblast, and there's 3 different kinds of trophoblasts. Long story short, she found that fusion RNA is expressed in the extravilli trophoblast that plays a role in the invasions and then penetrating of the uterine wall. Another thing that I want to, and because Luis told me that this is going to be videotaped and put on the web, my postdoc forbidden me to show the identity of this fusion RNA. I'm just going to call it fusion AB, but this is, if you're interested, I can tell you that about it later. But this is actually a bubble plot where we look at the GTACs, about 400 different individuals' blood samples, and then looking at how biased is the fusion RNAs being expressed in a sex-specific manner. And this particular one really jumps up, and it's only detected in female. And none of these, none of the samples or the male samples were positive for this particular fusion. So this fusion is a female-specific fusion RNA. This is experimental validation where we got, a bunch of the healthy donors from the lab, the poor postdocs and the students. And then there are females and males, and then you can see this particular fusion is expressed in a female-specific manner. But again, the parental gene are expressed both in male and females. So suddenly, the fusion RNA becoming a female-specific event. We were able to get some of the GTAC samples to validate, so they not only provide data, you can also get samples from them, and we actually look at it, and then all the female express this particular fusion RNA, and all the males are negative except one person. And that one person turns out to be a Klinefelter person, right? So XXY. So what matters is not the Y chromosome, is you have double X, right? If you have double X, you express this particular fusion. We actually got samples from our university hospital. I go there, every day, collect about 60 samples for my people to process. Last couple months, we got about 1,245 samples. When we look at it, and they see actually all the females, or 95% of the females are really high expressing this, and then majority of the males are not expressing this particular fusion. So that's consistent with our data. However, whenever you deal with clinical samples, you cannot 100% trust what's on the label of the blood tube. So some of these people, like we have to do a Y chromosome validation, and it could be errors. The nurse put in the wrong information. All these people are transgender. Who knows what's the percentage? Why is it, right? But we did Y chromosome validation, and some of these, like for instance, this male, it turns out to be a female. Stuff like that. So after that, we were able to correct some of that, and then show a even better segregation. However, these people are not 100% healthy, right? Otherwise, they wouldn't be in the hospital. So these outliers actually provide interesting information as well. And to get more statistically significant, we need 10,000, more samples. But from these few outliers, it seems like these are the person. Either they don't express it, if they're female, they don't express the fusion, or they're male, they express the fusion, tend to have circulating system diseases, and that's the reason they visit the hospital. So that's also the power of using clinical samples and having corresponding clinical data so we can connect the phenotypes as well. I won't go into the mechanism because it's going to take a lot of time, but I'm just summarizing. These two fusion RNAs are located on X chromosome, right? So it's not too surprising. And they're expressed in a female-specific manner, but what's interesting is this fusion RNA is made from the inactive X chromosome. So, female have two X chromosomes, one allele should be randomly inactivated, and there are about 10 to 15% of the X inactivated chromosome genes are escaping activation. So in the females, these genes are not really 100% inactivated in the inactivated allele. They do express a little bit, not as much, to the activated allele. So for instance, this A gene mostly expressed from the activated chromosome. B gene mostly expressed from activated chromosome. However, the fusion RNA is only produced from inactivated chromosome, and that's why it is a female-specific manner, because it's expressing from the inactivated allele. How do we know that? We actually take the breedings of 2 different mouse strains, and actually it's not strain, this is different species. They're different mouse that has a lot of polymorphism among them. So when you do the breeding, and then the female is actually delete heterozygous, which means they cannot inactivate their X chromosome. So all the inactivated allele is coming from Spratus mouse strain. So using that information, we can actually detect the fusion RNA, and it's all coming from the Spratus particular allele. So you know it's actually coming from the inactivated chromosome. All right, conclusion number 2. Chimeric RNA are widely spread, and the different tissues and cells and even different time points of differentiation can have different chimeric RNA profile. And certain chimeric RNAs can be tissue-specific or sex-specific, and at least some of the chimeric RNA are functional. I'm actually not showing the data. We have some published data as well to show that if you actually silence or force the silencing of certain chimeric RNAs, normal cell cannot live without it, or it affects a lot of physiological process. All right, so what does it all mean for cancer people? I'm trained as a cancer person, and so having all these chimeric RNAs that made by intergenic splicing could be a disaster for the cancer field when we try to just do a bunch of sequencing from a cancer cell line or cancer sample and jump into the conclusion we found some cancer markers. That actually is not correct. You're gonna really make sure that what you're finding at the chimeric RNA level, just by RNA sequencing, are truly the cancer-specific chimeric RNA events. On the other hand, it really expands the repertoire for biomarkers. So think about, we don't just have 20,000 genes, or we don't just have the 3 billion base pairs for point mutations, even doing the whole genome sequencing. That particular some chimeric RNA may actually work as great biomarkers or potential therapeutic targets for the cancer. So this is actually one of the cover stories that we had on Cancer Discovery, where we find a chimeric RNA that's made because of this C-splicing between neighbor genes, and it regulates prostate cancer proliferation. So again, I won't go into details of this particular story, but we rule out the mechanism of this particular fusion being a chromosome rearrangement or transsplicing. It turns out it's really a primary transcript going through the gene boundaries and getting to its neighbors. And so, and then alternative splicing occurs between these 2 neighbor genes. So you can also call it alternative splicing between neighbor genes. Again, long story short, we actually found this particular fusion RNA work as long non-coding RNA. So we call it long non-coding chimeric RNA, LNCC RNAs, and presented a bunch of evidence to support that. But it kind of worked as a scaffold to bring the PRC2 complex into the promoters of the P21 and a bunch of other downstream targets and work that way. So what's really interesting to us is this particular chimeric RNA level correlates with prostate cancer prognosis or Gleason score. So you know, this is log scale. You got a normal prostate, Gleason 6, 7, 8 to 10 metastasis. This chimeric RNA goes up. But the parental gene has no difference. So again, here I'm showing you one example of chimeric RNA correlates with disease progression, but the parental gene doesn't. So if you don't look for the chimeric RNAs, you actually will miss this particular biomarker as well. Conclusion number 3, misregulated transsplicing of C-SAGE. In this particular case, I'm talking about the read-throughs represent a new repertoire for biomarkers and drug target. And some of these chimeric RNA correlate with disease progression. We have satellite the pipeline, and we're working on many fields that we're not really familiar with. So, you know, muscle differentiation, stem cell, reproductive biology, and a lot of other things. So we are, eager for collaborations. So I'm probably gonna finish really fast. Good, save a lot of questions.
Yes. Thank you very much, Lee.
I haven't finished yet. Okay. So this is important. I had to acknowledge the past and current trainees and a lot of collaborators. in the world, and then my funding agencies, you know, including the Stand Up to Cancer, and now I had to plug in then this is the Lab..eLearning last Spring Festival in China. Chinese New Year, missing couple peoples, but to plug in a shout out for the Stand Up to Cancer, which actually is a grassroots foundation that organized by the entertainment foundation or entertainment business people. And so, you know, from the interactions between these people, really it really doesn't matter you're Selena Gomez or Katy Perry or Anna Hathaway, or your color of skin is yellow, white, or black, and you're singers or movie star or nerdy scientist. Almost, you know, everybody's life is going to be touched with cancer, right? So everyone in 2% is going to diagnose with cancer in their life, and your relative and friends or yourself may be diagnosed with cancer. So every life is going to be touched with cancer. And cancer really is the common enemy of Homo sapiens, and that's really the enemy we should be fighting, not really among ourselves. So that's really the message I want to give. And lastly, I want to plug in a shameless advertisement where I'm actually the editor of the particular specific volume book of chimeric RNA that's in the Method in Molecular Biology of Springer. And so if people are interested in contributing, just let me know and we can talk about that. All right, thank you very much. I think I finished really early, so we got more time to.
Thank you very much, Lee. Excellent work. Yes, I knew it. So this is the reason because he is giving a Caparica talk, okay?
Okay, very nice and very intriguing. So the signal with the Gleason score and in prostate cancer. So have you ever tried to use these chimeric transcripts as a signature for residual disease in patients that have been treated? Maybe they are to see whether they are coming back.
Right, so no, we haven't really used it as a residual disease monitoring, and it would be great if we can, and there's some evidence it may actually present in the urines, so you know, we're working hard to develop urine-based assays because that's non-invasive. This particular future RNA is not that highly expressed, so I worry about the future of using it as a very sensitive assay may not be great. I'm kind of giving this as an example because in the pipeline we have found a lot more chimeric RNA that seems to correlate with the, you know, the disease progressions, either the stage or Gleason scores. So I think some of those will be better markers than this one. But that's a good point.
So I was sort of interested in the distinction between sort of a cis mechanism and a trans mechanism for the chimeric RNAs. For your sex-specific event, was that like a trans splicing between two adjacent genes on the silenced chromosome, you said?
It is, yes. So it's coming from the inactivated X chromosome, right? But in this particular case, and that's a good point, is, you know, just the fact you have two neighbor genes making future RNA doesn't mean it's going to be a read-through or is it transplicing, right? Because the proximities may actually very well be transplicing. So in this particular case, we have evidence it's really a cis-splicing because we were able to detect the primary transcript from the, you know, the upstream gene into the downstream gene. And you know,
it's kind of a problem with transcription termination?
So that's also an interesting point is, you know, when you think about, and I may actually have some charts on that. Let's see. I save a lot of things. All right, so this particular case, right, like when you have the read-throughs or transcription cis-splicing between adjacent genes, we postulate that you have to meet three conditions, right? Your primary transcript has to be active. You have to go through the gene boundaries. You either have a leaky gene boundary or you have something allowing to go through. Your termination, you know, signals are weak or stuff like that. And then alternative splicings between exons of two different genes has to be encouraged. And we're actually finding evidence to support all the three factors. All three has to be met to have this kind of read-through event.
So that's interesting. This has been shown to happen in herpes virus infection.
Yes. So when you have the virus as well, it tends to go into the neighbors and stuff. So it's also very interesting there as well. We don't know how much it really contributes to the viral, but it's very interesting, and we're working a lot on the mechanism of this particular aspect.
Yes, sorry, you can talk, because there's a lot of people trying to ask. So more questions over there.
Yes, yes. Hi, I have a methods question, actually.
Yes.
So for the chimeric RNAs that must come from trans-splicing, did you exclude that they are not caused by template switching of the reverse transcriptase?
Right, yeah. So we are very aware of that particular potential artifacts, as well as, it may also, some of these may be real events by the template switching when you have a homologous sequence or short homologous sequence. In this particular case, we're looking at exactly exon boundaries that are less likely to be homologous recombination. And we actually look at, in the end, when you have these fusion RNAs, how similar are the end sequences or nearby sequences. We're not finding evidence of it's mediated by the short homologous sequence, right? But that's a good point.
Yeah, another question.
It's a really fascinating talk.
Thank
So is there any correlation between chimeric splicing and actual translocation events? Is it possible that the RNAs guide translocations?
Right. So that's one of my favorite topic is you're seeing, and that's the first part of my talk, is you're seeing exactly the final product of the chimeric RNA made by transplicing as well as the chromosome translocation in cancer. And so there's really two possibilities. One is maybe the same factor that contributing to the both processes are shared, right? So maybe the proximity of the two transcripts potentially are close by, and then active chromosomes, stuff like that. So the fact that you can make transplicing as well as you have a higher risk of chromosome translocation may be true, but not related. The other more interesting idea is maybe the chimeric RNA is serving as a mediator or template for the chromosome translocation. And there's evidence for that. There's Celia's data that's suggesting RNA, the micronuclei and macronuclei could actually serve as blueprint for your later on rearrangement. And there are also other data, especially by Storici from Georgia Tech University, that's supporting the chimeric RNA work as template for homologous recombination double-strand repair. So it's likely my student is actually working on that particular aspect.
Very interesting.
Thank you.
Yeah, more questions? There is another one. Yeah, go there.
Really interesting talk. I'm just wondering, in the cases where you see chimeric RNAs coming from different loci, do they correlate with their location when you look at high-C data? Are these actually genes which are clustering in the chromatin space in the same region, and then you detect these things?
Yes, so we are using some of these approaches. For instance, as you mentioned, using the ENCODE data and data mining, a lot of ENCODE data, we are actually finding particular histone modifications that correlating with these, as well as some of the polymerase transcriptions. Sorry, I have a lot of data I didn't really want to show, but I was worried about the timing. But yes, we are finding evidence of correlations with high-C data, and that's exactly the point that yes. But it's a little premature to talk about that at this moment. We want to gather more data on that, because unfortunately there are not many high-quality, high-resolution, high-C data out there. We're also doing our own.
In cancer cell types,
Use the microphone, please.
in cancer cell types, there's a lot of high-C data from Eric Lander's group.
Yeah, so
And
we're
so-
trying to avoid the cancer,
Right.
data because they could be chromosome translocations. So we're sort of more working on the normal, like ENCODE data, from the normal cell lines and tissues. So we're purposely avoiding the cancer data for this moment.
Any ideas you want to share on how you will test the role of these chimeric RNAs in normal development?
So that's harder question. Some of the data we publish, we are picking the low-hanging fruits, meaning that we are looking at the chimeric RNA expressed everywhere. So if they're expressed everywhere ubiquitously, they probably play some really simple fundamental functions. For instance, we silence it, using different approaches. The normal cell will just die. They can't leave it out, right? So they're really truly functional. But for the tissue-specific fusion RNAs, those are challenging questions. And And we're not expert in all the tissue types, and so it's very hard for us to go into these. But we're working on a few tissues, like the placenta. We're working on, some of these blood cells, like the female-specific one. We're really interested in what it do, right? What does it do in female? And I didn't show the data, but it's expressed in all the B cells, T cells, myeloid, lymphoid lineages. So it seems to share in all the lineages in a female-specific manner. And you think about it, like female has strong immune response than male, right? And that's why, most of the autoimmune diseases are female-specific. But on the other hand, if you have late-stage cancer, female benefit better because they have strong immune response. So, there's some research on that topic, like hormones plays a role, or some of the X chromosome gene plays a role. But we wonder, what is this future RNA maybe doing in this particular situation? So we're really, we're really actively working on that, and really also seeking collaborations on, what does it do in immunology as well. So, but it's a good topic.
Final question.
Have you considered to explain this phenomena in the context of a supraspliceosome in which the transcription goes on?
Every time I hear your talk, I think about that, but I don't exactly know how we're gonna study that topic. Super spliceosome. I would love to talk with you more about that topic.
So just for finishing, could you go to the first slide?
The first slide. Maybe he can do better.
Second slide then.
University of Virginia. You want to see that?
What is that?
That's a center building. It's called Rotunda. And it's really the replica of the Thomas Jefferson's home called Monticello, where you see on your nickel. So this is the University of Virginia's central building, and Thomas Jefferson was the designer and designed all these kind of things.
Like
He's
you.
the architect.
Congratulations for that. So finally, we clap and we acknowledge Lee. Thank you very much, Lee, for giving us this excellent talk. Thank you.